Hi, my name is Juliana Cruz, and in my microbiology class with Dr. Valkenaz, I isolated soil bacteria in order to screen for new antibiotic producers. Right now, there's an antibiotic crisis due to antibiotic resistance, and many bacterial diseases are becoming untreatable due to pathogens becoming resistant to treatment. The current pace of antibiotics is no match for the rapid evolution of resistance in these organisms. So antibiotics are typically produced by microorganisms, and there are a lot of microorganisms found in soil. So by collecting soil, you can contribute to helping solve this crisis. I had 10 isolates that I screened against E. coli, S. epidermidis, and B. subtilis, and this was important in order to find novel antibiotics to help with this crisis. So the objective of this experiment was to find and isolate antibiotic producers from diluted soil. And if there were positive producers, which later you'll see I did find one positive producer, secondary screens were completed to further investigate the properties and characteristics of the bacterium. To start with my methods, soil from California, PA was collected and diluted 5 times, so up to a 1.0 times 10 to the negative 5th dilution. From my last 3 dilution plates, spread plates were created, and from the spread plates, streak plates were created with 12 different bacterial types. A master plate was created from the streak plates, and the colon morphology was observed and recorded. I had, out of the 12 bacterium plated, only 10 grew, so this I just indicated with an X. So when you look at my pictures of the antibiotic screen plates, the X's just mean that no bacteria grew. For the antibiotic screens, these were run using safe relatives of pathogens B. subtilis on nutrient agar, E. coli on lysogeny broth, and S. epidermidis also on lysogeny broth. And the bacteria was picked and patched from the master plates onto each of these 3 plates. After this, 2 plates with EMB agar were used to plate 4 organisms using the streak plate method. 1 plate contained E. coli and B. subtilis split in half, one on one side, one on the other, and the same thing on the other plate, except with isolate number 4 and isolate number 3 from the master plates. Then 3 biochemical tests were conducted, and these tests consisted of a gelatin test on B. subtilis and isolate number 4, an oxidase test on B. subtilis, E. coli, and isolate number 4, and a catalase test with B. subtilis, E. coli, and isolate number 4. So if you direct your attention to my results, first off, we have the antibiotic production screen plates. And out of all the bacteria plated and screened, there was only 1 positive antibiotic producer on the plate that screened against B. subtilis. And you can see there, I have it circled in red, and you can tell by that clearing around the bacteria plated. So a clearing around a bacterium indicates a positive antibiotic producer. Next, we have my selective and differential media plate. And again, I used EMB agar. And EMB agar allows for the selection of gram-negative bacteria, and it differentiates between lactose fermenting and non-lactose fermenting bacteria. So first off, we have B. subtilis, and the B. subtilis had no growth on the EMB plate, and there was also no color change. So this indicates that the B. subtilis was gram-positive and non-lactose fermenting. B. subtilis is a gram-positive organism and does not ferment lactose, so the results support these facts. E. coli had a blue/black growth, and if you can see in the picture, it has a very metallic green sheen to it. And these results indicate the organism is a gram-negative lactose fermenting organism. So if there's growth, it is gram-negative, and the color change tells us that it is lactose fermenting. So in these selective and differential media plates, the B. subtilis and E. coli were used as controls. On the plate with the isolates number 3 and number 4, isolate number 3 showed a little bit of growth, but no color change. And this indicates that the organism is most likely gram-negative, non-lactose fermenting. And isolate 4 had growth as well as a light purple color. So this indicates it's a lactose fermenting gram-negative organism. These EMB agar plates are important because they allow for a lot of information to be known regarding bacteria, which is important for knowing how a certain bacterium will interact with humans. Lastly, we have the biochemical tests. For the gelatin test, B. subtilis turned the gelatin into a liquid. When the gelatin turns into a liquid, this indicates that the bacteria has the enzyme gelatinase. And the gelatinase hydrolyzed the gelatin. The tube with isolate number 4 still contained gelatin, so that means that no gelatinase was present or detected. Next up, we have the oxidase test. The oxidase test was run on E. coli, B. subtilis, and isolate number 4. As you can see in the pictures, there was no color change within these. And this indicates that there was no cytochrome oxidase present in the bacteria. The final test was the Catalase test, and all 3 had a positive result for this. And you can tell this by the bubbles that were produced. So all 3 produced bubbles. So this indicates that catalase was present, and the catalase broke down the hydrogen peroxide into oxygen and water. So these biochemical tests are some of the most important tests for microbial identification. It's a quick, cheap way to identify unknown organisms. So if I move myself up here, you can see that these are my references. I hope that you learned a little bit about this antibiotic crisis and enjoyed learning about the laboratory experiments and tests I conducted in order to try and find some novel antibiotics to help with the crisis. Thank you so much for your time and have a good day.